so that closed what we have to say about pyruvate as a precursor to alanine, valine, isoleucine, and leucine, as we saw. And now another important, and that's a glycolytic intermediate, right? So these amino acids are coming out. The basic building block is a fundamental glycolytic intermediate, the product of glycolysis in effect. Another very important group, as you will see in terms of cellular economy here, is the 3-phosphoglycerate group, and in particular serine. Okay, so 3-phosphoglycerate will give rise straight to serine, and from then on we go to glycine and cysteine. And we'll see why that's important. It has received quite a bit of attention lately due to its involvement in cancer. So how do we go from 3-phosphoglycerate to serine shown here? It's an extremely straightforward reaction. First, we have a redox reaction with an AD and an ADH. So we oxidize this hydroxyl carbon on 3-phosphoglycerate to a ketocarbon, right? And once you do that, then you have a transaminase. Another name for transaminase is actually aminotransferase. Sorry, yeah. And you just go straight from 3-phosphopyruvate, okay, to 3-phosphoserine. Okay, you change. So the point of the first reaction is to generate the keto acid, and then with a transaminase, you make the amino acid, and that at this point will be 3-phosphoserine. All right. So then again, this is a very important transamination reaction, very common. Glutamate is a key participant in all these transamination reactions. We've covered this before. And once you make 3-phosphoserine, then all you have to do is go in with a phosphatase, clip that phosphate group up, and you got serine. So it's a very straightforward biosynthesis, going from 3-phosphoglycerate to serine. But what has been quite important is this generation of alpha-ketoglutarate here during serine biosynthesis. Now, hold on a second, because I have the window open and there is a cardinal that is just driving me bananas. Hold on. Okay, sorry about that. So we eliminated nature's noises here, but that could be disturbing for this. So anyway, so back to serine biosynthesis, the steps are straightforward. And what became very interesting here is, based on the recent studies that I will show you in a minute, is I want to point out the generation of alpha-ketoglutarate as a standard transaminase product. You have the amino acid gets converted to the keto acid, and vice versa. The keto acid here, 3-phosphopyruvate, is converted to the amino acid. Well, it turns out that serine biosynthesis is essential in cancer. The cells rely enormously on these three reactions. Why is that? So the surprise came from some measurements of the fluxes in this pathway. So this is glycolysis, right? 3-phosphoglycerate gives pyruvate, and then with fermentation you get lactate, as we discussed many lectures ago. In the first, this is the first reaction, okay, and this is where 3-phosphoglycerate in serine biosynthesis, it gets diverted to make towards serine synthesis. Now it turns out that about 9%. So if you ask how much of this carbon flow gets diverted towards making serine, okay, it turns out 9%. So that's almost a tenth of the whole glycolytic flux in breast cancer cells is used to make serine ultimately, okay? And the answer, why is that important? So serine is very important by itself, as you will see in the next slide, because it's a precursor to many important things. But in addition, it turns out that this transaminase that we're talking about, so this one here, okay, so you get the first reaction of the dehydrogenase, you convert the 2-hydroxyl group to keto group, and then the transamination, this is the phosphoserine aminotransferase or transaminase, will take glutamate as the amino acid, and that's the alpha keto acid that participates, the compound that we have here, the phosphocyrene, the precursor to phosphocyrene, to generate phosphocyrene, that's amino acid, and generate the equivalent alpha keto acid from glutamate, which of course alpha ketoglutarate. Now it turns out that 50% of the alpha ketoglutarate used in cancer cells is derived from this reaction. So the real requirement in cancer cells for this pathway, for the most part, arises from the generation of alpha ketoglutarate, the all important 5-carbon intermediate in the TCA cycle that we've been talking about. And this is really what this does to the cell. It provides the source of alpha ketoglutarate. As surprising as it may seem, a simple transamination here is giving half of all alpha ketoglutarate, at least in these growing cells, these rapidly proliferating breast cancer cells. And in addition, of course, what you get is a lot of serine, right? I mean, when you go, when you go back to this reaction, from phosphocyrene, you're going to get serine with a simple phosphatase at the last step. And serine is in turn used enormously. Okay, how? A lot of serine goes into making phospholipids, ceramides in particular. So those are the lipids that were discussed. Okay, and again, why would you want to do that? Well, if you're a growing cell and you want to make a lot of membranes, that needs a lot of phospholipids, a lot of phosphocyrene. So this is really why this is useful. In addition, from serine, so this is the PS here, but you can also make the ceramides. From serine, we get, in one step, we get glycine. That's part of the 1-carbon metabolism, actually. So the 1 carbon of serine is lost. It gets onto folates, and this is where the folate, most of the folates get their 1 carbon, as we saw in the previous lectures, which then they can move on to other things, let's say making methionine, for example, for the methyl cycle. And that leaves glycine, of course, and from serine you also get cysteine, as we saw in the cysteine biosynthesis. And these are basic amino acids you can feed into all kinds of protein synthesis. So with serine, you satisfy, lipids, making membranes. You can satisfy making, a lot of serine goes into making protein. These are very abundant amino acids, glycine and serine. So many proteins have lots of serines and glycines usually. So you can see why serine is such an important sink of carbon coming out of glycolysis, right? And if you add to that a lot of the 1 carbon of serine goes into folates and all these methylations in the cell, you can see how serine also provides that entry, the 1 carbon unit that goes into folates to make